My name is Kay Tai, and I'm an assistant professor here at the Picower Institute for Learning and Memory at MIT in the Department of Brain and Cognitive Sciences. My lab is interested in understanding the neural circuits that underlie motivated behaviors. Here, in this study, Edward Nye, a graduate student in my lab, found that an inhibitory projection from the lateral hypothalamus, or the LH, going to the ventral tegmental area, or the VTA, is important for not only disinhibiting VTA dopamine neurons, but also increasing behavioral activation. Dopamine is a neurotransmitter that has been linked to a number of functions, including motivated behaviors. Our study shows how stimulating the inhibitory GABAergic input from the LH to the VTA increases the motivation for an animal to interact with salient stimuli, which can depend on the context of their environment. For example, if food is available, stimulating the projection can cause feeding. If a novel object is present, that may increase novel object investigation. If a social agent is present, the same stimulation can cause an increase in social interaction. This study provides new insights into this very important circuit in the brain and helps us better understand the inputs that can regulate motivated behaviors.
Hi, I'm Edward Nye, a graduate student in Kay Tai's lab at MIT, as well as lead researcher on this project. And for our experiments, we used transgenic mice to help us express pteridopsin 2, a protein that makes neurons light sensitive, and specifically GABA cells in the LH. And then we could implant optic fibers over the VTA that allows us to shine blue light to activate specifically the direct projection from the GABA cells in the LH to the VTA. And to test whether the stimulation was actually preferred or avoided by our mice, we placed them into a chamber where one side was associated with stimulation, and we measured the amount of time they spent on either side. And what we found was that animals actually preferred to be on the side where they were getting stimulated. In addition, they were also very willing to work for the stimulation by poking their nose into a port, where each time they poke, they get 1 second's worth of stimulation. And this told us that activating the GABAergic LH to VTA projection was actually positively reinforcing. Even more interesting is that when we place these mice into a cage with an intruder mouse, either a juvenile male or adult female animal, stimulating the projection actually caused the mice to increase their interaction with the new animal. Combined with our previous work showing that stimulating this projection can also cause feeding behaviors, this told us that activating this one projection could actually generate very different behaviors depending on the context or the environment.
I'm Caitlin Vanderweel. I'm a graduate student in the lab and a co-first author on this paper. So at this point we were wondering, how does activating an inhibitory connection from the lateral hypothalamus to the ventral tegmental area generate this seemingly appetitive motivated behaviors that we are observing? Since we tend to think that activating rather than inhibiting the VTA is important for reward and reinforcement. So we decided to examine downstream dopamine release from the ventral tegmental area to one of its major outputs, the nucleus accumbens, in order to see how these LH inputs were influencing dopaminergic activity. Of course, we know that dopamine signaling in the nucleus accumbens is crucial for motivation and reward-related behaviors, so we hypothesized that somehow this inhibitory LH projection to the VTA is increasing downstream dopamine release. So I used a technique called fast-scan cyclic voltammetry, which uses a carbon fiber to measure electrochemical changes associated with fluctuations in dopamine release in the nucleus accumbens, while simultaneously activating this inhibitory input from the LH to the VTA. And what we found was that activation of this projection does increase dopamine release in the nucleus accumbens, which did support the kinds of appetitively motivated behaviors that we were observing.
The key to finding the last piece of puzzle was the inhibitory GABAergic neurons in the VTA. For inhibitory projection to the VTA to cause dopamine release, our hypothesis was the signal had to pass through these GABA neurons in the VTA. And to test this hypothesis, we used a technique combining a redshifted opsin called crimson R with a genetically encoded calcium indicator called GCAMP6M. This allowed us to stimulate GABAergic LH to VTA projections with yellow light and use blue light to activate the GCAMP, which allows us to see the activity of GABA neurons in the VTA. When we did this, we found that activating the GABAergic LH to VTA projection caused a decrease in activity in the GABA neurons of the VTA. Essentially, stimulating the GABAergic LH to VTA projection turns off the neurons that turn off the dopamine neurons, essentially causing dopamine neurons to turn on. And just like grade school arithmetic, 2 negatives do make a positive. So the next time you want to take a sip of your favorite soft drink or hang out with your friends, remember that this projection in your brain is probably firing and causing dopamine release.